So how glycogen is synthesized? There is cell, and inside the cell we have a part of the glycogen molecule. We know that glycogen consists of glucose molecules that are connected mostly in a linear fashion by alpha-1,4 glycosidic bonds. And when person intake a high carbohydrate meal, glucose level in the blood increase, and high amount of glucose molecules through a GLUT transporter enter into the cell. In most cells in human body, glucose is trapped inside the cell by the enzyme called hexokinase that phosphorylates glucose into glucose 6-phosphate, and glucose with the phosphate group cannot leave the cell. Also, we have to know that liver tissue, in addition to hexokinase, also has glucogenase that also phosphorylates glucose to glucose 6-phosphate. So once glucose is trapped, cell can use glucose 6-phosphate for its own purposes. Initially, glucose 6-phosphate is converted to glucose 1-phosphate. The problem here is that we have to transport glucose 1-phosphate to the glycogen, and this transportation occurs by UDP molecule. UDP picks up glucose 1-phosphate with formation of UDP-Glucose. Important to know that UDP-Glucose is a well-known glucose donor. Basically, UDP-Glucose molecule provides glucose for all metabolic processes that require glucose. For example, for glycosylation of proteins, or in our case for glycogen synthesis. So this UDP-Glucose molecule comes to the glycogen and transfers glucose to the last glucose molecule in the glycogen structure. And the major enzyme in glycogen synthesis, glycogen synthase, binds this glucose molecule by a 1,4 glycosidic bond. So in this fashion, glycogen molecule can be enlarged in a linear fashion. So to explain this in more details, when UDP delivers additional glucose molecule, glycogen synthase binds this new glucose molecule by the first carbon to the fourth carbon of a last glucose molecule in a glycogen structure. As a result, 1,4 glycosidic bond is formed. Then, for example, another UDP-Glucose molecule comes to the glycogen, and glycogen synthase binds this another glucose molecule to the fourth carbon of a glucose in a glycogen structure. And in this fashion, glycogen molecule can be enlarged further and further. But at some point, this horizontal glucose chain becomes so large that it requires branching, because you cannot store huge amount of glucose molecules just in one line. And glycogen branches are created by the specific enzyme that is called branching enzyme. When UDP delivers glucose to the glycogen, branching enzyme binds this glucose to the glucose that is already present in a glycogen structure by alpha-1,6 glycosidic bond. And the specific feature of alpha-1,6 glycosidic bond that it binds glucose in a vertical fashion that results in formation of a brand new branch. And now this branch can be enlarged by adding of a new glucose molecules by a usual 1,4 glycosidic bond. So to illustrate this, when UDP delivers glucose to the glycogen, now branching enzyme connects this glucose molecule by the first carbon to the sixth carbon of a glucose that is already present in a glycogen structure. As a result, alpha-1,6 glycosidic bond is formed. And now when UDP delivers additional glucose molecule, glycogen synthase connects this new glucose molecule by a usual alpha-1,4 glycosidic bond. And in this fashion, this branch enlarges further and further. As we see, because glucose molecules are binded mostly in a linear fashion, the major enzyme in glycogen synthesis is glycogen synthase. Because of its importance, glycogen synthase has to be regulated. The two major factors that regulate glycogen synthase function are insulin and glucagon. Logically, because glycogen synthesis is an anabolic process, insulin stimulates glycogen synthase and glucagon inhibits glycogen synthase.